Hello everyone, my name is Akriti Bhandari. Today I am going to discuss a topic of microbiology, that is gram staining. In this topic, I am going to discuss about the introduction first, then principle behind it, reagents involved, mechanism, processor, then result, after that application and limitations. Let's begin with the introduction first. The gram staining is the most fundamental technique used in microbiology, which is used to classify the bacteria into two broad categories, that is, gram positive and gram negative bacteria, on the basis of their cell wall composition. This technique was developed by Hans Christian Gram in 1884. It is a differential staining. As I told you earlier, we will get the classification of both the bacteria on this basis. Some of you might be thinking, why do we use gram staining technique? The answer is, it helps in identifying bacteria quickly. It gives us initial information about the infection. It helps doctors to choose the appropriate antibiotics regarding the bacterial infection. It is one of the first steps in microbiological diagnosis. Now we will discuss the principle behind the gram staining technique. The principle of gram staining is based on the structural and chemical differences in the bacterial cell wall. The cell wall of bacteria is made up of peptidoglycan layer. Gram-positive bacteria will have thick peptidoglycan layer, whereas the gram-negative bacteria will have thin peptidoglycan layer. Now it's essential to understand the reagents used and their roles in the process. First one is primary stain, that is crystal violet. It is a basic dye and generally it stains all bacterial cell. Crystal violet is a basic dye or a primary stain which penetrates the cell wall and cytoplasm. Next is iodine solution, which is act as a mordant. It forms a crystal violet iodine complex, and the role of it to enhance the binding capacity of primary stain with the bacterial cell. It helps to trap the dye inside the gram-positive cells during decolorization process. In the absence of mordant, the primary stain may wash out from all cells and the differentiation would fail. So it is an important step. Next one is decolorization, or we can say use of alcohol. 95% ethanol is used as a decolorization agent. It is a differential step that separates gram-positive from gram-negative bacteria. What actually happens? Gram-positive bacteria having thick peptidoglycan layer, which shrinks in size, and due to having crystal violet iodine complex, the all cells remain purple in color. But gram-negative bacteria having lipid outer membrane dissolves and crystal violet iodine complex washed away and it become colorless during decolorization process. It is again a most critical reagent in this process. The last reagent used is safranin, that is a counter stain. It is a basic red dye. It stains all the decolorized cells. The gram-positive cells already retain the violet color, so they are not affected, but the gram-negative cells will contain or will retain this safranin dye and become red in color. Now we will perform the procedure. How will we perform this experiment or the staining procedure in the laboratory conditions? Step 1, we have to prepare the smear. For the smear preparation, we have to take a clean grease-free glass slide and we have to take a drop of sterile water and place onto the slide. Using a sterile loop, we have to transfer a small amount of bacterial culture and spread it to form a thin and uniform smear, even smear. Allow the smear to air dry completely. Next step is heat fixation. Pass the dried slide quickly through the flame 2-3 times. The main purpose of heat fixation is to kill the bacteria and to fix the cell to the slide to preserve the cell morphology. But one point should be remembered, we have to avoid overheating, otherwise cell will be distorted. Next step is we have to do the staining. First is primary staining, that is with the crystal violet dye. We have to flood the smear with crystal violet, leave it for about 1 minute, then gently rinse with water. And what we'll get, all bacterial cells will appear violet in color. Next, we have to apply mordant or iodine solution. Flood the smear with iodine solution, leave for 1 minute, then rinse gently with water. Now iodine acts as a mordant. It forms a crystal violet iodine complex inside the cell. This complex is larger and more stable, which will help gram-positive cells to retain the crystal violet stain. Next is decolorization. That is a critical step of this staining. Now we have to add 95% of ethanol for about 20 seconds. Immediately rinse with water. Now what will the result? The gram-positive bacteria having thick peptidoglycan will remain violet in color due to having the crystal violet iodine complex. But gram-negative bacteria having lipid outer membrane, it will be dissolved and crystal violet complex will be washed away and it become colorless. While performing decolorization process, one thing should be remembered that over or under decolorization can give the false results. Next is counter staining. Now we have to flood the smear with safranin for 30 to 60 seconds. Wash gently with water or blot it dry. Now what happened? The gram-negative bacteria will get pink or red stain while gram-positive bacteria remain violet in color. Now let's know about bacterial cell wall. The bacterial cell wall is the outermost layer of bacterial cell. It will maintain the cell shape, it will provide the mechanical strength, it will prevent the osmotic lysis, and it mainly contributing to the staining properties, especially in the gram staining processor. The major component of bacterial cell wall is peptidoglycan, that is murine, which is responsible for its strength and rigidity. Now, peptidoglycan is a mesh-like polymer made up of glycan chains, alternating units of N-acetylglycamine and N-acetylmuramic acid. Gram-positive bacterial cell wall having thick peptidoglycan layer that is 20 to 18 nanometer. It contains teichoic acids and lipoteichoic acids. No outer membrane present and outside it, and it has low lipid content. Examples of gram-positive bacteria are Staphylococcus aureus, Streptococcus pyogenes, Bacillus subtilis, Lactobacillus. Gram-negative bacterial cell wall have thin peptidoglycan layer that is around 2 to 7 nanometer and single or few layer. It also possesses an outer membrane which is having high lipid content. Gram-negative bacteria having a unique feature of outer membrane, that is it contains lipopolysaccharides. It contains lipid A endotoxin, core polysaccharides, O antigen, which will having a antigenic specificity, porine proteins and phospholipase and lipoprotein in excess amount. Examples of gram-negative bacteria are Escherichia coli, Salmonella typhi, Pseudomonas aeruginosa. Now we'll discuss with the application part. Application of gram staining. It is used for rapid preliminary identification of bacteria in patient samples such as blood, urine, sputum, and other fluids also. It also helps to detect the infection like urinary tract infection, pneumonia, meningitis, provides immediate information before culture results are available. It will help the doctor to choose the appropriate treatment for the patients. It will help in the study of bacterial morphology, the shape of the bacteria, whether it is a cocci shaped, bacilli shaped, spirali, or the arrangement, whether it is in the form of cluster, chains, or in pairs. And it is also used in research and training purposes. It is widely used in microbiology laboratories for studying bacterial structure, demonstrating staining techniques to train the students. Staining is also used in the food industry, water testing, and in pharmaceutical products to identify the bacterial contamination to ensure the safety. Although gram staining is very useful and widely used technique, it has several limitations also, as it is not applicable to all the bacteria. Some bacteria do not stain well with gram stain, such as Mycobacterium, require acid-fast staining, Mycoplasma, which lacks a cell wall. Therefore, gram staining cannot be used for all type of bacteria. Another limitation is that it always requires a fresh culture. Old bacterial cultures that is more than 24 hours may lose their ability to retain stain properly. This can give false result also.